Chapter 2 is going to focus on the chemical context of life. We're going to first take a look at matter, and more specifically elements and compounds. All organisms are made of matter, anything that takes up space and has mass. Elements are one form of matter that can be combined with other elements to form compounds. Elements cannot be broken down into anything simpler, either by chemical reactions or by physical changes. Compounds are going to consist of two or more elements that have been combined together in a fixed ratio and will have characteristics that differ from the elements that were used to comprise that compound. There are 92 elements that are naturally occurring, and there's some debate as to whether there's more, depending on if you want to account for some of the ones that are present in minute quantities or some of the ones that are going to form as a result of other radioactive elements decaying. But of the 92 that typically are assumed to be naturally occurring, 20 to 25% of them are absolutely essential for living things. Carbon, hydrogen, oxygen, and nitrogen, and you see them at the bottom of this slide. They make up over 95% of living matter. The majority of what's left will consist of calcium, phosphorus, potassium, and sulfur, and there are some other trace elements that are required, like iron. So what you see down there at the bottom are the electron shells representing those four elements. Hydrogen has 1 electron, oxygen has 8, nitrogen has 7, and carbon has 6. Why they have a different valence number than they have electrons, why they have a different number of electrons in that outermost ring as opposed to their total number of electrons, has to do with the number of electrons that are present in their outermost shell. You may remember from when you looked at electrons in middle school, how the first ring, the first energy level, could take up to 2 electrons. Hydrogen, not having the 2, only has that 1 valence shell, and it has a space for 1 more electron, hence the valence of 1. Oxygen fills that first valence shell with 2 of its 8 electrons, and that leaves 6 behind. So you can see the dot structure underneath there, and you see how 4 of the electrons are going to be written with pairs of 2, and then you have 2 that are solo. If you had 2 more electrons, you would bring that shell up to a filled shell, hence the valence of 2. And we see the same thing with nitrogen with its 7 electrons, 2 in the first energy level, 5 in the outermost. 5 plus 3 gives us 8. And then finally with carbon, carbon has 6 electrons, 2 in the first energy level, and so it's going to have 4 in its outermost ring. So this is just looking at the relative percentages that we find of those elements. Again, oxygen is going to make up the vast majority of your body's mass. Carbon's going to be right behind it. Hydrogen and nitrogen, definitely greater percentages than some of the other elements, but not nearly as much as oxygen and carbon. Then we see the other elements that are comprising the roughly 4%. And then you see those trace elements listed down there at the bottom that are only needed in very, very small percentages. So how elements are going to differ from one another is going to depend on what is making up their individual atoms. The atoms are going to be your simplest unit of matter that is able to retain the properties of the element, but we cannot isolate atoms from an element and be able to visibly see them. We would have to look at them under a microscope. But every element is going to have a unique number of atoms because every element is going to have a unique number of protons in its atom's nucleus. So atoms are composed not just of protons, but also neutrons and electrons. Protons positively charged, neutrons neutral, electrons negatively charged. The protons and the neutrons are going to make up your atom's nucleus. The electrons are going to be localized outside the nucleus in an electron cloud. And the mass of a proton and neutron is pretty comparable. So when you look at their masses, they're roughly the same, and you can measure them in kilograms or grams, or you can measure them in what's called a Dalton. So there's a picture of, I was going to say it's helium, yeah, because we've got the 2 protons and we've got the 2 neutrons, and then the electron cloud is dispersed outside the nucleus, and you see the 2 2 electrons present for helium. I gave you a periodic table with the papers that I handed out today. I didn't hand out today, the papers I handed out the first day of school. And because I knew you were going to have some questions involving elements, and I wanted you to have a reference to be able to look at. Atoms of different elements are going to vary based on their subatomic particles, not just necessarily their protons, but they can also vary in terms of their neutrons. Your atomic number, which is always going to be present on a periodic table, tells you the number of protons that are present in a nucleus. The mass number tells us the protons plus the neutrons that are present. You cannot get the mass number strictly off of the periodic table. You can approximate the mass number off of the periodic table, especially for elements that are have smaller atomic numbers, because the vast majority of their average mass is typically due to one isotope being present in greater quantities. So isotopes for elements are going to differ by their neutrons, so they'll have different overall masses, but they'll be the same element because they have the same number of protons. Radioactive isotopes are ones that are not stable. The ratio of protons to neutrons is either too high or too low, and they will go through decay spontaneously. They can give off helium particles. They can give off particles that have the approximate charge and mass of an electron, or the approximate mass but a positive charge that's comparable to that of an electron. Or they could just give off large amounts of energy. When these are going to play a big role with biology, because we can use these to kind of trace things with medicine. Sometimes you will be asked to, you'll be given an IV that will contain radioactive isotopes, so they can use it as a tracer to check and see how things are working through your body. We can also use it to date fossils. And in this particular picture, you can see the bright yellow color. That is an indication of rapid cell division. So you could trace like sulfurs or phosphorus atoms that might be a part of cell division. And when you see that rapid cell division occurring, you see those bright colors, that might be an indication that there's an abnormality that needs to be looked at further. So talked a little bit about energy so far. Energy is a capacity to cause change. You can have stored energy in the form of potential energy that's due to the location of that particular form of matter, or just its inherent structure, the energy that's stored in the bonds that are holding that particular form of matter together. Atoms, electrons are going to differ in their potential energy depending on where those electrons are at in comparison to the nucleus. The energy levels, those circles that I kind of talked about a little bit earlier, are going to be an indication of the potential energy. The further the electrons are away from the nucleus, the more potential energy they're likely to have. So here you've got an analogy of a ball falling down. In terms of as it falls down, it's going to take in more and more energy. So to have electrons move outward from the first shell out to the third shell, they're going to require fair amounts of energy to be able to do that kind of shift. But for the electrons to fall back down to where they would normally be found in their ground state, they're going to release energy. So the periodic table is going to show us the electron distribution for each element's electrons. Again, that first energy level is going to show you the electrons that would be present in the core level. You typically are going to fill that up after you get past helium. And then electrons that are found in subsequent rings are going to also be core electrons until you get to that outermost ring. That outermost shell is where you have your valence electrons, the electrons that are further separated from the nucleus. They are the ones that are going to be participating in bonding. And they are the ones that are going to determine what types of interactions any particular atom will have with other elements. When you have a filled valence shell, so again, after the first energy level, which can hold up to 2, all the others for our purposes are going to be holding up to 8. When you have filled that valence shell, you no longer have a reactive element. It is considered to be chemically inert, and that's what we see with our noble gases. They have a completely filled energy level, so they are very unlikely to be involved in chemical reactions. If we look at your periodic table, you will see that the elements in group 1 have one valence electron. So if they lose that one valence electron, they get to revert back to a filled energy level. And that's part of why your alkali metals are so reactive. Whereas if we go over to the group just before the noble gases, the halogens in group 17, fluorine, chlorine, bromine, and iodine, you'll see in this particular picture they are one shy of having their energy level, their outermost valence energy level completely filled. They will beg, borrow, and steal to get that extra electron so they have a filled energy level. So your halogens are your most reactive nonmetals. You should be comfortable with drawing those types of diagrams for a given element based on the information that's provided in terms of if it's an atom or if it's an ion, and being able to determine the number of protons and the number of electrons. So orbitals, again, are generalized regions where we're likely to find electrons at. We find them approximately 90% of the time, and every energy shell or every energy level will have a set number of orbitals in which electrons can be placed, and a maximum of 2 electrons can be placed in any given orbital. So how molecules are going to form is going to be determined by the type of bonds that can form between those individual atoms. I talked a little bit before about how the goal is to get a filled valence shell, to either get to 2 if you're hydrogen or helium, or maybe lithium or beryllium, the ones that are just past 2, or to get to that magic 8. So elements will lose electrons, elements will gain electrons, elements will share electrons in the form of chemical bonds so that they can form these stable molecules and get their filled valence shells. Covalent bonds are going to result from a sharing of electrons, so there's a physical connection between the two. And when they are sharing electrons, they get to count for both atoms. So in this particular case, hydrogen has 1 valence electron, carbon has 4. And so if 4 hydrogens combine with 1 carbon atom, each of the hydrogen atoms gets their second valence electron, and carbon gets the remaining 4 valence electrons. And that allows us to form methane, which is a fairly stable molecule. Molecules are going to consist of 2 or more atoms that are held together by covalent bonds. Notice I did not say they had to be different atoms. Your study of chemistry, you most likely ran across your diatomic elements. Brinkelhoff, nitrogen, oxygen, fluorine, chlorine, bromine, iodine, and hydrogen. Those are examples of molecules. They share their electrons covalently. Sometimes they do it with a single bond, sometimes they do it with a double, and other times they'll do it with a triple. When you have a single covalent bond, you're sharing 1 pair of electrons between the 2 atoms. When you have 2 pairs of electrons connecting the atoms together, you have your double bond. And then in the case of acetylene down there at the bottom, you see that it has 3 pairs of electrons. You have to use all the valence electrons that each element contributes to create these molecules, and you have to find a way to share them such that every element that wants to get 8 is able to get its 8, and that hydrogen does not take on more than 2. There's some more specifics that we get into in AP Chem, but for your purposes, that should be sufficient. Sometimes you will see the dots that are representing the electrons that are being shared written with lines. When you see those lines, we're dealing with structural formulas, and then we can simplify that even further by just writing the chemical formula of the atoms that have been combined together. So here you see examples of the electron distribution diagrams for 2 molecules of the same element, hydrogen and oxygen, as well as 2 molecules made of different elements, water and methane. We see the Lewis dot structure where we see just the valence electrons that are being shared between the atoms. And we also see the structural formula showing the number of bonds by the lines. And then you can see a more of a 3D representation of what those molecules would look like. Electronegativity plays a pretty big role in determining how atoms are going to connect together. Some elements want electrons more strongly than others, And so if an atom is more attracted for electrons in a covalent bond, it's going to draw the electrons more towards it. So in this particular chart, this is an Allen scale, and we typically use the Pauling scale. We don't typically use the Allen scale, so my apologies for that. Noble gases typically are not assigned electronegativity values on the Pauling scale because they already have all the electrons that they need. They already have a filled valence shell. But if you look at fluorine, you see that even on this chart, it has the highest electronegativity of the non-halogens that are present. Fluorine is your most electronegative atom on the periodic table. It is the one that is going to draw electrons to it more strongly than anything else. If we look on and make a diagonal and we go down towards the bottom left-hand corner, we see that cesium and francium have some of the lowest electronegativity values assigned. They're only over their the last filled energy shell by 1 valence electron. So rather than taking on up to 7 more electrons, they are much more willing to give up their 1 valence electron to create a stable bond. In a covalent bond, because sharing is taking place, you are more likely to have atoms form covalent bonds that have similar electronegativities. They do not have to be the same. And we typically see that with our nonmetals and occasionally our metalloids. If you have a diatomic molecule that is made of the same elements, they have formed a nonpolar covalent bond. When oxygen forms a bond with another oxygen atom and makes O2, you have a double bond that's going to result from 2 pairs of electrons being shared, but they're going to be shared equally. There's no reason for one oxygen to want electrons more than the other. In a polar covalent bond, you do have one atom that has more of an affinity for electrons than another. We see that with water. Oxygen is much more electronegative than hydrogen. So in water, the bond between H and O, the electrons, like tug of war, are going to be pulled more towards oxygen. Oxygen wants those electrons more strongly than hydrogen. And when that happens, there's a little bit of a shift in that electrical charge, and that causes one side of the molecule to be slightly negative and the other side to be slightly positive. Ionic bonds are going to form when you physically have an electron be transferred. You have electrons move from cations to anions, and when you have these electrons move, you no longer have just a neutral atom, you have a charged atom, also known as an ion. Cations are your positive ions, anions are your negative ions, and ionic bonds are going to form when you have these oppositely charged ions come together. We see these more with elements that have very different electronegativities. There's not nearly as much of an interest in sharing. Something really wants to give up electrons and something else really wants to gain them. And we see that here in the example of sodium chloride. Sodium has 1 valence electron. Chlorine is shy 1 valence electron. So chlorine, being much more electronegative, takes the electron away from sodium, leaving behind a sodium cation and a chlorine anion. And the pull between them is so strong that it keeps these two ions together as a unit. So we also refer to these compounds as salts. And that's how you would see them often in nature in their crystalline form. However, when they are added to water, the ions are able to separate because water's polarity, that you have the partially negative oxygen atom and the partially positive hydrogen atoms, they're able to surround the two ions and kind of pull them apart from one another. Because of that, ionic bonds definitely have way more energy stored in them. But when you have an ionic compound that's been dissolved in water, you have the ions physically separated far enough from one another that your covalent bonds, the ones that are permanently present, that are being shared, are going to be a little stronger. So again, I know that's kind of an oxymoron. There's a lot of debate with chemists and biologists about why ionic bonds are much stronger in chemistry and not as strong in biology. The biggest thing I can tell you is that with ionic bonds in biology, when they are added to, when you have compounds like that added to water, that is enough of a disruption between the two ions that they no longer have to stay necessarily together. Whereas with a covalent bond, they have to still stay together. We haven't touched on hydrogen bonds yet. Hydrogen bonds actually are not a true connection between atoms that make up a molecule. They are intermolecular attractions, so they are attractions between molecules either of the same type or two molecules that have similar polarities, but they basically pull um, molecules to draw towards one another. So we see that a lot with water, and we're going to talk about that a lot in our next chapter. The intermolecular forces kind of help substances retain the state that they're in or their shape so that they're able to stay relatively intact, but they require much, much less energy than your covalent bonds or your ionic bonds. Okay, we see hydrogen bonds between nitrogen, oxygen, or fluorine. They have one of those really super electronegative elements needs to be present, and they will form hydrogen bonds with other molecules that contain hydrogen. So here we have ammonia, NH3. Nitrogen being super electronegative will be drawn towards the hydrogen end of water. And then if you have additional water molecules, that oxygen might be drawn towards the hydrogens that are present in ammonia. So you can make lots of these little connections, and those connections have to be overcome for something to change state. We don't tend to run into fluorine so much in living cells. We tend to run into more of oxygen and nitrogen as a result of hydrogen bonding. So one other type of intermolecular force that we're going to spend some time on are van der Waals interactions. So van der Waals are going to come about when you have molecules. They could be polar, they could be non-polar, but electrons are in constant random motion, and as a result of that, sometimes the electrons might end up asymmetrically distributed. One side of the molecule might be more positively charged, the other side might be more negatively charged. And these are short, instantaneous shifts, and they don't last very long. However, if you have one of these spots, it can cause another shift in an adjacent molecule. And if you have enough of these form, you will have van der Waals, or what we talk about in AP Chem, London dispersion forces that take place. And so these are going to play a role in some of the other properties that we see with substances. And here you have an example with a gecko's toe hair and being able to walk up a wall. Okay, that you're able to find attractions because of these asymmetrical distributions that are super, super short-lived. In biology and chemistry, it really doesn't matter. The shape of your molecule is definitely going to influence its ability to function. How its shape is going to be determined is based on those valence orbitals that are filled, how many pairs of electrons are present in that molecule, and how they're going to be able to work together to form a stable three-dimensional shape. We see with covalent bonds that the s and the p orbitals that are present with that outermost valence shell, that's where you're going to find those electrons at, can create areas of 2 electrons around the central atom. It can create areas of 3 areas of electrons around the central atom, and it can create 4. It can go further than that. But because of that, you will see shapes that are more linear, or you'll see shapes that are more in the form of a triangle. Sometimes you'll see shapes that are even in the form of a tetrahedral. Biological molecules are able to both recognize and interact because of these specific shapes. They can only fit in certain ways. And so you can have similar molecules that will be able to work in such a manner because they have something about their shapes that is able to be recognized. So endorphins are one example of this. If you look at the structure of a natural endorphin, you see the box in black. And then if you look at the structure of morphine, you see that there's some very close similarities between the two. And so the receptors on our brain cells that recognize these natural endorphins are also going to be capable of recognizing morphine. And so this would be an example where two different molecules can mimic the same impact on your cells because they both are going to be able to fit that receptor appropriately. Chemical reactions are going to result in the forming of chemical bonds, but they're also going to result in the breaking of chemical bonds. Your reactants, your starting products, and your reactants, your starting materials, and your products, your final materials are going to have to be converted between one another. Photosynthesis is one example here. Use the bonds between this carbon and oxygen and CO2 have to be broken. The bonds between H and O and water have to be broken to form a glucose and then to form oxygen molecules. I said first that reactants and products are going to have to work together to make one another. All chemical reactions are capable of being reversed, going from reactants to products or products to reactants. Some reactions are more inclined to go backwards and forwards to show that equilibrium when we see a mixture of both reactants and products present. And then there are many, many chemical reactions that are only going to go in one direction, or only going to pretty much favor the forward direction or the reverse direction. Just because of the thermodynamics that are involved, it doesn't make a whole lot of logical sense for them to move in both directions. In biology, we see a lot of equilibrium because we have a lot of reactions that will take place in multiple steps, and we need to be able to reform what we've made previously. So we're going to see a lot of that context as we get further on this year.